The overall goal of this procedure is to unveil interactions between proteins and DNA in the chromatin of Arabidopsis cells.
This method can help answer key questions in the plant biology fields, such as which genes are regulated by a given transcription factor, or what histone modifications are associated with the transcriptional status of genes.
The main advantage of this technique is that through the cross-linking step, it is possible to fix and capture changes in chromatin organization occurring during plant development.
Though this method can provide insight into protein-DNA interaction in whole seedling of Arabidopsis thaliana, it also can be applied to different plant organs and adapted to other plant species.
Demonstrating this procedure will be Dorota Komar and Alfonso Mouriz, two PhD students in our laboratory.
After growing Arabidopsis according to the text protocol, collect 1.5 grams of plant material in 50 milliliter tubes and keep them on ice during cross-linking. Then, use a laboratory burner-heated needle to make little holes in the lids of the tubes. Add 40 milliliters of 1-xPBS and 1.08 milliliters of formaldehyde to the tubes. Screw the lids on the tubes and place the tubes in a desiccator. Vacuum infiltrate for 10 minutes. Then carefully release the vacuum to prevent churning up the solution. After mixing